Okay, students, we've spent a lot of time talking about proteins, important proteins like transport proteins like myoglobin and hemoglobin. And then we also took a lot of time to talk about enzymes. And now we're going to switch gears and look at another type of important macromolecule in biochemistry that are called lipids. So there's the main functions of lipids will be energy storage, membrane structure, and signaling. And so what this group is, interestingly enough, it's going to have some more variety in how it looks. But what they all have in common is that they are water insoluble. So this second part here, I'm going to switch up colors today. That means that they're water insoluble. And that's what they're going to have in common as we look more closely at their structures. Okay. The other thing that's important is that they're going to be amphipathic, meaning that they're going to have a region that's polar, but another region that is nonpolar and hydrophobic. So it's going to have a hydrophilic part and a hydrophobic region. If we put lipids into water, they tend to form what are called micelle structures, which is that the nonpolar parts tend to attract. So this would be the center part here. So those would attract through van der Waals. And then the outside would be the polar part would be attracted to the water. Okay, so let's start off with our most simple of lipids, the most famous, I believe, which are our fatty acids. And fatty acids essentially have a long hydrocarbon chain. So there's our long hydrocarbon chain. And then it has the polar head. Now, one of the things that we've been talking about a lot is that the carboxylic acid, the COOH part here, that part would be deprotonated at a physiological pH. So as we keep moving forward, you're going to see that a better representation of that would have been deprotonated with the rest of the chain. Okay, some terminology with regards to fatty acids. When the hydrocarbon chain are all single bonds, they're said to be saturated. So if we look at the structure shown here, which is stearic acid, which is an 18-carbon fatty acid, that is saturated, meaning that there's only single bonds. And then there are unsaturated fatty acids that contain one or more double bonds. So this is oleic acid. It's a 18-carbon fatty acid. And then the counting here in the notation, so this would be carbon 1, 2, 3, 4, 5, 6, 7, 8, 9. So if you notice, the notation for oleic acid has this notation, and that's telling us that the double bond is on the 9th carbon. Okay, the other thing that's important about this double bond that we'll see in a few minutes is they always have the cis configuration. So if you recall from organic chem, cis means that the carbons here are on the same side. And that's going to be an important discussion for us later. Okay, here's another view of this. Here has the space filling view. And then here's the idea, because of the pKa of that carboxylic group at pH 7.4, that when we see pictures of them later, they should be shown as deprotonated. Okay. Here, a couple of very important things. If you note in the saturated fatty acid, so that one's saturated. If you look at the space-fill model, it forms a rigid chain, whereas in the cis configuration, there tends to be a bend or a kink in the chain. Okay. Okay. This will be an important chart, and this will be present for you on your exam. So this would be given on your test, or a variation of it. So you're not memorizing this. What I want to show you is a couple things. Here are some very common saturated fatty acids. The more commonly referenced are palmitic and stearic acid. Those come up a lot. The one with 16 carbons and 18. Notice the abbreviation of 16.0. That means there's no double bonds. And then here's a really good structure of these. That is a more condensed view, but you want to picture they have a very long hydrocarbon chain. And then again, these at pH 7.4 would be COO minus. Okay, notice what happens is that the more carbons they have, you see an increase in the melting point, and that's because you have stronger van der Waals forces. Okay, so stronger intermolecular forces. Okay, and then the unsaturated, these are all pretty famous. Oleic, very famous, linoleic. If we take a look at these, you'll see that the first two are an unsaturated fatty acid where the double bond is in carbon 9. And then the ones here with more than one double bond, those guys would be polyunsaturated. Okay, so we want to take a look at a little bit more in terms of how this will affect their properties. So one of the things we're going to start getting into is the idea that function is affected by structure. We're saying it a little differently. Structure affects function. Okay. The fluidity of the fatty acids will decrease as the chain length increases, meaning that their melting point will increase. And the fluidity of the fatty acid decreases as the number of cis bonds decreases. So what is this going to really mean for us in real life is that our saturated fatty acids have stronger intermolecular forces because they can get closer together, and that these guys tend to be solids at room temp. Whereas our unsaturated fats, what will happen there is, if you notice here, their melting points are lower, right? And so these guys tend to be liquids. Now, these are liquids, but in real life, we would call these oils. And remember that that's a property of lipids, is they do not want to interact with water. They're not water soluble. So we know that oil and water do not mix. I'm going to go back one more thing on our fatty acids. For those of you that are in nutrition majors, one of the things that is very important as nutrition majors is to really understand these fatty acids in relation to heart disease. So saturated fats, because they tend to be solids, they tend to accumulate on the heart. And so saturated fats would be things that we think of, or high amounts in like butter or animal lard. Whereas unsaturated fatty acids would be the oils. So when you have different oils like canola oil, olive oil, they tend to be a blend of these different unsaturated fats. And for the body, nutritionally, this is a much better choice because they tend to not accumulate on the heart. The other thing that's really important to note is, let's take a look at linolenic acid here. I'm going to switch colors on my pen. If I count, now the counting for the carbon chains on these was from the carboxylic acid. So that's the numbers that we get. So when we're counting from that end, if you notice, we'd have 1 carbon, 7 would be 8, 9. So that's carbon 9, right? And that's where the double bond is, that's the notation and on the abbreviation of linolenic acid. But there's another thing that comes into play, and I know as nutrition majors, you've heard a lot about this, is some of these are omega-3 fatty acids. So linolenic 1, 2, 3. Okay, and so that is an omega-3, meaning that we are counting from the end. That is what omega means, the end. And omega-3 fatty acids have been well studied over the last 20 years or so in regards to improving improving heart function and decreasing statistically heart disease. And one of the ways that they found that out was to look at pockets in the world, pockets of people that live to be very old. And one of the things that they observed about those groups of people is that their diets were very rich in omega-3 fatty acids. And usually like with certain fish that contain a high amount. What American diets tend to be low because we don't necessarily tend to eat a lot of fish as a whole. And so they supplement a lot of our food with omega-3s. There's also omega-6 fatty acids, which would mean when you count 1, 2, 3, 4, 5, 6. So this one also has a double bond on the 6th position from the end. Now, what happened at one point was that our foods became a lot more processed starting in the 1960s and 70s. And so the European way, for example, of eating bread was to use these oils that were blends of unsaturated. You've probably been to Italian restaurants where you dip it in the oil. Solids like butter are hard to spread on a piece of bread. So what food chemists did was they took unsaturated fats and added hydrogen to them in a process called partial hydrogenation. To create a substance that was in between an unsaturated fat and a saturated fat. So somewhere kind of in between an oil and a solid. And then that was where the idea of margarine was created. The problem was that during that partial hydrogenation process, instead of forming bonds, and I'll go back, instead of forming these cis fats, instead was what was formed in partial hydrogenation, or what are called trans fats. And very quickly, scientists realized that trans fats increase the risk of heart disease. And trans fats were being used a lot in fast food restaurants to fry food, used a lot in frozen foods. And so the government then had to step in and do more severe restrictions on them within our food. Okay, our next topic is our least favorite topic, and that are fats or triacylglycerides. And what these are fats that how we store fats within our body. So an important cell type that we're going to keep looking at are adipose cells. Those are what we call fat cells. And fat cells are cells that store fat. And one of the important ones, or I should say, the type of fat that's stored in them are triacylglycerides. Now, I'll be honest with you, I'm going to abbreviate that as TAG. When I was in school many moons ago, we used to call these triglycerides. And so I can't help myself but call them that, but most newer texts will call them triacylglycerols or triacylglycerides. So I'm going to try to erase that so you don't get in that habit of calling them that. Okay, these are storage molecules for fats, and what you should see as we look at them is they have a theme, and their theme is as follows. They have a glycerol backbone. So here's the backbone that they have. And then they have a linkage to 3 fatty acids. Now what we're going to do in our Zoom lecture is we're just going to practice writing these structures. So we're going to have a little bit more hands-on. So we will come back to this and we'll practice writing them because it can be hard for students. What I want you to notice about them, and you can really see it in the space fill model, is what a bulky structure this is and what an enormously nonpolar structure this is. So this is super nonpolar. And as a nonpolar structure, it excludes water. And that is an important little fact about them, because what it means is when they're stored in our adipose cells or fat cells, we can store quite a bit of it because it packs in there excluding and removing water, so you don't have that water weight that's added to the cell. Okay, another type of lipid are waxes and soaps. So here's a typical wax. They have a long fatty acid chain. So here's our friend oleic acid again. And then it's bound to another long chain. So a lot of times waxes are used as protective coatings for plants and animals. And again, excluding out water, right? And then there's the soaps. Soaps tend to be formed by fatty acids reacting with strong bases to form a salt. And so when we take a look at the way a soap works, is it's going to have the long hydrocarbon chain again. And that's going to attract the grease through London dispersion forces. And then the polar head will attract the water. Okay, I'm going to click on this picture. I don't want to go through this right now. What our next category is, and we will be practicing this more in the Zoom lecture, is a very famous type of lipid called a phospholipid. And what a phospholipid is, these are our main components in cell membranes. So this is our cell membrane component. Okay, let's take a look at the backbone. So I'm going to use my fancy highlighter again. The backbone is what I'm highlighting here. So what they'll all have in common, it's not quite glycerol. It has 2 carbons. I'm going to go back to my pen. It's going to have 1, 2, 3 carbons, right? But the third one down here is going to have phosphate. So here's phosphate. And that's where the word phospho comes in. That's why they're phospholipids. Okay, and then the carbon 1 and 2 is going to have a fatty acid attached. Now, this one shown here, phosphatidic acid, it's our most simple phospholipid because you see this, that's an H right there. Now, a phospholipid can have different groups, so it doesn't have to be H. So we have this, can be other groups. And so what other groups it can have are shown here. So here's our phosphatidic acid. That's our most simple phospholipid. But then if you notice, you can have phosphatidyl ethanolamine. You can have phosphatidylcholine, phosphatidylserine, phosphatidyl inositol. And this might look very intimidating, but what I want you to realize is if they start substituting these R groups on, what you're doing is you're creating a charged polar head on the end of that phospholipid. Okay, one other type of lipid are sphingolipids, and they don't have a glycerol backbone. What they have is what's called a sphingosine backbone. So let me get the highlighter. Here, this whole thing is sphingosine. Okay, so it has a definite chain on that carbon 1. So you see the carbon 1 that's highlighted here. Oops, sorry. That is set in stone. It has this C double bond group, CH212, CH3. So that's always the same in a sphingolipid. It always has that backbone. What will be different will be this amino group, NH2, will form an amide linkage to a fatty acid. Okay, and then adding to the complexity here, let's take a look at this OH. This would be one of our more simple sphingolipids that's called a ceramide. It's going to have this OH group. Okay, but that hydrogen can be substituted out for other charged groups. So in this case, this sphingolipid is substituted out with a phosphate and also choline. So now you have what's called a phosphosphingolipid. Okay, now why are these important? These dominate our nervous system. So they dominate in nerves and in the brain cell membranes. So the sphingolipids or sphingophospholipids are very important components of cell membranes in the brain and in the nervous system. Another thing that also dominates in brain biochemistry are what are called glycolipids. And what these guys are, if you notice, the backbone, here's our friend sphingosine. So that's the sphingosine backbone. Here would be the amide linkage to a fatty acid. And then attached to this CH2 group is a sugar. And so this is what's called a glucocerebroside. Okay, and so that cerebroside would have one sugar. And then you can have very complex carbohydrates attached. And those guys are called gangliosides. So when you have more than 3 sugars, those are gangliosides. Let's see if there's a pic on the next one. So things start to get really crazy looking on these structures. So in this case, believe it or not, here's our sphingosine backbone. Okay, that's the sphingosine highlighted with the highlighter. And then you have a whole bunch of sugar moieties on here, glucose, galactose. And you also, what's very common on a ganglioside is to have sialic acid, which is this complex looking group right here. These tend to dominate in brain biochemistry. And so they are important in the nervous system. Lastly, another important type of lipid are steroids. And steroids have this particular fused ring structure. And one of the most important steroids that we talk about is cholesterol. So right here is the structure of cholesterol. You see the backbone as shown here. And we will see in our next discussion the importance of cholesterol in the cell membrane. Okay, so to recap, we started to learn about fatty acids. We also looked at our storage molecule for fats called triacylglycerides or triacylglycerols. We also looked at phospholipids, which are important in cell membrane biochemistry. We also looked at sphingolipids, which are important in cell membranes of the nervous system and brain. And then we looked at glycolipids and steroids. Their structures are a little intimidating, but what they all have in common is that they are dominated by nonpolar groups. We did see on the phospholipids, though, that they're very amphipathic, meaning that part of them is nonpolar and the other part's polar. So what we're going to do is, I'm hoping you're watching this as a preparation for the Zoom discussion we'll have, where I go back through and we practice writing some of these structures.